Stitching a gene into a piece of DNA is critical for producing a protein like insulin. Let's take a look closely at the elements needed to make a recombinant expression vector. What cell line we choose to host the production will of course influence how we design our vector, but expression vectors usually share 5 common features. These are an origin of replication, a gene of interest, a promoter sequence, a terminator sequence, and a gene encoding a selectable marker. These elements are organized on a circular piece of DNA called a plasmid. This is our vector. Let's look at each element in more detail. The first is an origin of replication. This sequence is a region where the process of DNA replication begins. Plasmids are native to many prokaryotes, so the cell's natural machinery will replicate both its genome and the plasmid. This allows the cells to continue carrying the plasmid through many generations. It also allows us to produce many copies of the vector in bacteria for transfection into other hosts. The second element is the gene of interest itself. For a single protein like insulin, the gene is stitched into the vector using enzymatic ligation. The gene has to be inserted in frame so that the gene aligns naturally with the framework of the plasmid itself. This ensures that the transcription machinery can read the individual codons correctly. The third element, the promoter sequence, allows efficient binding of the transcriptional machinery to the plasmid, including an RNA polymerase. The promoter is positioned at the 5' end, or the front, of the gene of interest, and will lead to the transcription of the biologic into mRNA, which is subsequently translated into protein. In the simplest design, the vector will instruct the cell to produce our protein of interest intracellularly. These elements can be arranged in many ways. Sometimes additional elements may be inserted in between them to improve productivity or to make recovering the biologic easier. The fourth element is a terminator sequence. This section of DNA is designed to limit transcription to the gene of interest on the plasmid and is positioned at the 3' end of the gene. Without a terminator, the transcriptional machinery can keep going past the end of the gene, leading to variants of your product. The terminator is transcribed at the end of the mRNA encoding the biologic. It helps stabilize the mRNA itself by forming secondary loop structures at the 3' end. This additional stability reduces the degradation of the mRNA and ultimately promotes higher productivity. The fifth element is a gene called a selectable marker. Not all cells exposed to the vector will take it up. A selectable marker allows you to select for those that do. This gene confers a survival advantage to cells with the vector. Exposing all cells to whatever toxin or additive that the gene protects against will allow the cells to survive. In this way, you can select for only the cells that took up the vector and kill the rest. So organizing these 5 elements into a plasmid make it possible for us to introduce a heterologous gene for a biologic into a host cell. Allow that host to replicate the DNA sequence and express the protein, and allow us to select those cells that carry the vector. These transformed cells can now produce our biologic of interest.